further ado, I would like to introduce you to our speaker this week, Dr. Dimitry Gorsky, and he is joining us from Buffalo, New York, where he's a fish biologist with the Lower Great Lakes Fish and Wildlife Conservation Office. So I'll let him tell you a little bit more about himself. Take it away.
Thanks. Hi, everybody. I'm calling you from my attic. I really wish I could be up in Vermont seeing you all in person. These virtual things are not easy, so I appreciate you spending the time listening, and hopefully I'm engaging enough that you don't get bored to sleep. Thank you, Dr. Tarleton. Do they call you Emily? Me and Emily are friends from high school, actually. It's a very funny story. So it's really nice to see her again and have this opportunity to get this invitation. So let me share my slides.
If you want any background dirt on Dimitry, I got it.
Oh, no, don't ask. You don't want to know.
Second part of the presentation?
That could be included in your paper for extra credit. All right, so I'll share this, and I guess before we get started, okay, there we go. I can open everybody up. It's working. So where was I gonna go with this? So I'm a fish biologist with the U.S. Fish and Wildlife Service. It's a government agency, and you know, there's a lot of environmental jobs at many different levels, right? There's the government agency, there's state agencies, then there's nonprofit like Sea Grant and the Nature Conservancy. There's a lot of opportunities for environmental jobs, and I just happen to be at this federal level position. And there I'm a fish biologist, and what that sort of means is we monitor and study fish populations to make sure that they're healthy, that they're growing, that maybe they're sustainable if people are fishing for them. A good example of that is in Lake Erie. There's a very heavy sport fishery and commercial fishery for walleye. And so it's part of our job to make sure that there's enough fish out there that they can grow and they can reproduce. And so we do a lot of different pieces of that kind of work. Sort of as a background here, I just kind of put some numbers down so that I could remember. I went to undergraduate school in Rhode Island at Roger Williams University. I originally thought I wanted to be a marine biologist, and I don't think I was wrong, but my direction changed eventually. It was a really great school. It was a private school, and it cost a lot of money. I wish I went to a state school, something that was maybe more affordable, but it was a really great experience. Once I left there, it was really difficult to find a job. I was doing a lot of seasonal work. I was doing waitering, I was doing jobs that were completely outside my field. But eventually in 2002, I got a big break. I got hired in Maine to work on an Atlantic salmon project. And when I say that this was probably my 500th or 600th application that I submitted, I don't think I'm exaggerating. It took a lot of effort to get that one break. And it was originally just a 6-month position, but I got up there and just, busted my butt to work really hard and do the best I could. And they kept me on. Every 6 months they would renew it and they would keep me on. And so that was sort of, the turning point for me. I took the project that they hired me for and convinced the principal investigators to turn it into a master's program. And so that's where I started grad school in like 2002, 2003, I think. And I got a master's in ecology and environmental science. And what I was studying was Atlantic salmon coming back from the ocean would swim up these rivers, and they'd have to swim through multiple dams. They'd have to get past all these dams to get to habitat where they could reproduce. And so I was marking them and tracking them and finding out, where they were hitting roadblocks, dam blocks, what was stopping them. And so that was a really amazing experience. Maine is beautiful. I mean, you guys are in Vermont, so what am I talking about? It's beautiful up there too. But so that was that work. And while doing that, I ended up meeting a professor who then offered me an opportunity for a PhD. And again, I was having a really hard time finding the next level job. And so I went after that and ended up getting a PhD in wildlife ecology. It's still working with fish, but at that university, they called it wildlife. They included fish in that umbrella. And so up there for that project, I was doing a lot of work on inland lakes up in the state of Maine. And so again, a wonderful experience. And when I was finished, the same routine, lots of applications, trying to get this next step. And luckily for me, I got an opportunity here at this office. And again, it wasn't ideal. I came down here, it was a job they only had for 4 years. It wasn't a full permanent job. And so I came down here and worked really hard and did the best I could do until the opportunity came where I got hired as a permanent staff. And so when I talk to, people early in their careers and students like yourselves, I think a lot about the stories that I heard when I was in your shoes. And it was, you got to be lucky, you got to know the right people. It's always sort of these same stories. But in the end, I think that everything, this is like sort of my vision of it, is that you have to make yourself available, and it has to be your best self, right? You're putting yourself out there, you're meeting people, and you're working hard. And when those opportunities arise, you just step 1 foot forward and you're in that door, right? And so, a lot of people say it's luck, but it's only lucky for the people that really set themselves up, right? And so there's a lot of work that you can do to let luck find you. I don't know if that really hits home with any of you, but that's sort of the way that I look back on my path into this position. So I guess I wanted to finish this intro on, I have this nice sunny picture of me with a fish here. When people see me doing my job out in the wild, they're like, man, you got the best job. You're the luckiest ever. Well, sometimes it's true and sometimes it's not. This is me working one day and it was just snowing like crazy. And, there's no fishermen out looking at me telling me I'm lucky to have that job because they're not out there, right? So we work in all types of weather, all sorts of conditions. But so what I wanted to share with you all today for current topics in science is a topic that's pretty important in the Great Lakes. I don't know how many of you have ever seen or heard of lake sturgeon. They're really an amazing fish. They're actually native to Lake Champlain. So there's I can't remember I want to say the Lemoy, is that the name of a river in Vermont? That river has a sturgeon population in it. It's really small, but the state is working on restoring that population. Here in the Great Lakes, they're kind of a big deal. They were nearly wiped out, and we're working really hard to recover them. And I'm going to share some of the research and thinking that my coworkers in my office have done to develop sort of this recovery plan that we use that we're working on in the Lower Niagara River. So sturgeon across the Northern Hemisphere are in the family Acipenseridae. That's the sturgeon family. They're very easily identifiable, recognizable. They have these really cute whiskers. Their mouth is on the bottom. They sort of eat like vacuum cleaners. They swim around and eat stuff off the bottom. They look very much like a shark. And in fact, they're cartilaginous, just like sharks are. They also have a heterocercal tail, which means it's longer up top than it is on the bottom. And that's really a characteristic you see in some sharks. what's really cool is they have bony plates, and they have, 4 lines of them. This one goes down their back. They have lines on their sides and then on their belly. A lot of sturgeon look very similar to one another. There are some that are longer and skinnier, but you can really tell pretty quickly that it's a sturgeon by looking at it. And they call it a living fossil because when you look back in the fossil records on sturgeon, they look pretty much the same as they do now. Their ancestors don't look very different. The lake sturgeon is the biggest fish in the Great Lakes, but it's not the biggest sturgeon in North America. There's Atlantic sturgeon that are anadromous. That means they go in the freshwater and into salt water on the East Coast. And then there's white sturgeon out on the West Coast. And so, these are very big fish, but Atlantic sturgeon will get 12, 13 feet, and white sturgeon will get even bigger than that. So they're really amazing creatures. So like I was saying, the lake sturgeon's big. It's the biggest in the Great Lakes, biggest fish in the Great Lakes. It'll grow over 8 feet long and over 300 pounds. What we typically see are the fish that I try to include a lot of pictures here. They're usually around 6 feet or under 6 feet long, and they're rarely over 100 pounds, but these are still really sizable fish. Another characteristic of them that's really cool is that they live a really long time. The females will live over 100 years old, up to 150 years old. And the males will live 50 or 60 years. I skipped over this bullet here. The record that we've seen was a dead fish that washed up on the shore of Lake Erie, and it was 7'11", over 300 pounds. And they estimated that it was a female that was about 86 years old. So really just big, big fish. What's going on here? There we go. Their reproductive behavior is really interesting too. They take a while to mature, kind of, similar to us, right? They're not sexually mature right away. The males will take 8 to 12 years before they're sexually mature, and the females take longer. Sometimes, mostly over 14 years, sometimes up to 30 years before they have their first reproductive event. Once they are sexually mature and start reproducing, they don't reproduce every year. A lot of other fish do reproduce every year. Some fish even reproduce multiple times in a year. And so males will typically take off a year, 2 to 7 years, and the females take several years off, 4 to 9 years off. And so what this means is, like, in a given year, if you're looking at a group of spawning sturgeon, you're only looking at a small percentage of that population, because the rest of the population is out in the lake eating and growing and sort of rebuilding their energy stores. And so it makes it really hard to study. You never know, one of the questions we have is how many are out there, right? And so at any given time, if we're out there looking, we're only looking at a small segment of that population. So it takes a while, a long study, before you can really figure out some of those questions. And so here's a general life cycle. We'll start here with the adults. They feed in the Great Lakes in shallow waters. A lot of people, when they hear their bottom feeders, they think they're really deep. We typically see them 30, 40, 50 feet deep. We do have evidence that they're deeper than that, but they're certainly not in like the deepest spot of the lake. In the spring for spawning, they return to the river they're born in, and they spawn in really high flow waters. I have a picture and video later. And the eggs are sticky. And so the eggs come out, they get fertilized, and they stick right to rocks, and they hold tight there. The eggs incubate for like a week or 2 weeks after they're spawned, and then they'll hatch, and the larvae drift downstream, and they sort of come out of the water column where the river calms down. And then the juveniles eventually move out into the Great Lakes. They stay in the river for a little while, up to 4 years. And they grow pretty fast when they're little. and then they move out to the Great Lakes and become an adult. This is a diagram that I like to share when I think about life cycles, because once, you know, a fish once fish are adults, they sort of continually migrate through different habitats in life. And so, you know, we just talked about here the spawning habitat, and, you know, that's up in the river. The fish go up there not every year. So it's a habitat that they spend some time in through their life. But then after that, they have a migration out to feeding habitat, or the juveniles hatch and they go out to nursery habitat where they can grow. So you have to have this arrow, this migration corridor available for fish to move. From the feeding habitat, then you often have an overwintering area where they move to. It's usually a little deeper, because in water, in lakes, when it gets cold, the warmest water is often the deepest water. And so they'll find an area like that where it's warmer and they'll feed. And so again, you have this back and forth between this habitat, and then you have to have a migration corridor here back to the spawning habitat. And so I mentioned this because dams pose a big issue for sturgeon. When you put a dam in a river, you block these two corridors, right? And you essentially isolate the spawning habitat, and they may or may not be able to get there. I just realized I forgot to mention, if you guys have questions, throw them in the chat and we'll get to them at the end. I wanted to mention that in case you had some good questions. Okay, why isn't it moving? So, sorry, give me a second here. So historically, here's a diagram of the Great Lakes, and historically, all these green dots represented populations that had a river where the fish had access and could go up and spawn, and everything was happy. You know, they were healthy, reproducing populations. And so currently, all these red squares represent populations where they're extirpated, which means they're locally extinct. There's no evidence of any juveniles or adults in those river systems. And there's actually some green dots on this figure, which I challenge you to even find, because those represent large intact population. And there's only like 3 of them. The black dots are smaller remnant populations, and that's where we, as our agency, are focused. We're trying to save those populations, make sure that they're growing, and at the same time, we're reintroducing populations in all those red boxes. So there's a lot of work here to do. This is, you know, quite devastating. And so why have they declined? One of the biggest issues was commercial fishing, which I'll show you in a little bit. Another issue, like I explained with the habitat transition diagram, is habitat loss. If you put dams up, you're essentially blocking them from ever being able to reproduce. And so that can be really devastating. Water pollution is always an issue. The toxic chemicals sometimes interfere with reproduction, and invasive species. Sea lamprey are known to predate on them, and round goby is an invader that is known to eat a lot of eggs, and so that may have an impact as well. But going to the commercial fishery, this is a graph from some old data, and so it goes from the year 1875 to 1995. On the y-axis here is the commercial catch in thousands of pounds. And so each one of these lines represents one of the Great Lakes fisheries. And you can see, if you look at this green line, the peak was right around 1885 maybe, and they were harvesting over 1 million pounds of sturgeon a year. And so looking at this 20-year spread from 1880 to 1905, they really harvested these fish a lot. This shows anywhere from 200,000 a year to 1 million a year. And so I left off a couple lakes and I'm gonna add them now. It's the same figure, the same data, but this is now including Lake Erie. So this was the big peak that we saw at 1 million. So Lake Erie was harvesting 5 million pounds a year at one point. And so you can just imagine the impact. If an average fish is 100 pounds, that's 500,000 fish, or I'm sorry, 50,000 fish a year that they're pulling out of the system. And so that was really devastating. And what you can see is once that fishery really did its damage, there was no recovery. There was not a lot of fish showing up again. Here's an example just to show the habitat destruction. This is an old picture of the Detroit River, where this is really good spawning habitat for sturgeon. But what they did is they dried up the river and blew this up with dynamite to make way for shipping, right? So now they have a channel going right through this beautiful sturgeon habitat so that ships can get by. And so that clearly has some big impacts on sturgeon. Oh yeah, here's another picture of them just pulling out all of that dirt to make that channel. So that's sort of the bigger Great Lakes stuff. And I'm gonna zoom in a little bit and talk about the Niagara River. Hopefully some of you have been to Niagara Falls. It's an amazing site. It's a really beautiful waterfall. Here's a picture of the Horseshoe Falls. That's only half of the fall. There's another waterfall over here. This little piece is just an island. But the Niagara River starts in Lake Erie near Buffalo, the city of Buffalo, New York, and it flows north over Niagara Falls and then into Lake Ontario. Niagara Falls almost perfectly cuts the river in half. And so we call this section up by Buffalo the upper river, and the section down here the lower river. And so there's been some signs of recovery. Even, back in the late 90s, my office started a volunteer sighting survey. So we were asking fishermen, anybody recreating on boats. A lot of the stuff came in from fishermen. We even had a lot of scuba divers that were scuba diving in the river that would report sightings of sturgeon. So all of these dots in the river represent sighting data where they said that they were seeing fish. So we knew, we have good information that the fish are there. And so we didn't know much else though. That was about all we knew. And so back in 2010, 2011, we started to do a really focused population assessment. We wanted to really learn about these critters out there. And so what I'm going to share with you is really all the work that we've done, and it's mostly taken place on the lower Niagara River. We are starting to do a lot of this same work now up on the upper river, but these studies took a really long time to do. So here's Niagara Falls again. This is sort of where the upper river starts, and you can see from Niagara Falls down to Lewiston, it's really narrow. In this section, it's really rapid. You can't take boats in there at all. They have jet boats that go through there for tourists. And it's I think it's a pretty fun experience if you ever get a chance, but you really can't get up there to do any research. So that whole section of the river has been sort of isolated from the work we've been able to do. But once you get downstream of Lewiston, you can see it's a lot wider. And once it widens out, it really kind of slows down and you can actually get some pretty calm, manageable spots to work. And so we had a lot of research questions, and this is me and my buddy just looking dazed and confused and asking, how many are there? What are their ages? Are they reproducing? We know there's a lot of adults. People are seeing them out there. Are they reproducing? What are they eating? And what habitats are important to them? Where are they spending their time? I'm thinking again about where's their feeding, overwintering habitat, things like that. And so we started sampling for them. We needed to catch them in order to start answering these questions. And so these projects are really cool because they combined a lot of new technology that I'll talk about with some older technology. And the older technology are these set lines. This is the way the commercial fishermen used to catch them all the time. They would take a really long line and they'd clip baited hooks to them, and they would just sink the line on the bottom of the river or the lake, and they would just have it going there overnight. And so we did that. We would set them off the side of the boat like this. We'd come back the next day and hopefully have several sturgeon on there for collection and study. When we pulled them in, we'd collect a bunch of biological data, their length, their weight, their girth. We'd take a spine sample for aging, which I'll talk about in a second, and we would take a tissue sample for genetics. And so we wanted to see how the population was genetically also. And then this last bullet was a big one. We wanted to estimate how many were out there. And so we used a statistical model that looks at how many fish you mark, and then when you come back, how many of those marked fish do you catch? And you do this over and over again, and eventually you can run this statistical model that tells you how many, estimates how many are out there. And so every time we caught a fish for this model, we would tag them with a pit tag. And it's essentially the same thing like animals get, those ID tags that are injected. And so every time a fish shows up, we scan it and we'd have an ID and we'd know everything about that fish. And so that's how we kept track of everything. And so this figure is the beginning of some data. I have a lot of data to share, and hopefully it doesn't get too boring. I'll try to keep it light, but what this is showing is over the years of sampling for sturgeon in the lower river, how much effort are we putting, how many are we catching for the same amount of effort? And so you can just sort of, if you squint your eyes just right, you can see that we've gotten better. We're catching a lot more fish with less effort. And so, this early study, they were catching 10 a year, 16, 22. And so by the time we were done, we were catching, 200, over 100 fish each year. And so we were able to really identify where they were spending time and have a good method to pull them in. So here's, again, some more data. I'll walk you through it. So right over here, this is each year that we did the survey. We actually ran the survey to 2018, but I didn't include it because this gets too big. So I left it like this. Here is the number of fish we caught each year. Okay, and so you can see it's variable, but we were catching around 100 a year. This section here shows how many we are recapturing, right? So this number 1 here was tagged. It was one of these 28 fish that was tagged in 2010, and we caught it in 2011, right? And so then this one is another fish that was tagged in 2010 that we caught in 2012. And so when you go down to the end here, you can see this last column shows that every year we're catching 100 fish, and about 10 or 11 of them were tagged in a previous year. And so this all goes into that statistical model where we just keep, the goal is to get as many recaptures as we can. And that's how you make the best model. And so down here, we collected 1,125 individuals, and we had a total of 90 recaptures. And so we put those numbers into this CJS model, and it estimates that every year there's a 92% chance that the fish will survive. So that's really, in fisheries world, that's a really high number. 92% of them will survive each year. And then this capture probability tells us how good we are at capturing the fish. So if a fish is there, we have a 3% chance of catching it. So we're not really good at catching these fish. But ultimately, the whole goal was to generate this population estimate. And so we think that there's around 6,500 fish in this one population. And so that was great news. We knew they were there, but we certainly had no idea there was 6,500 of them there. That's a lot. So I mentioned age estimating. we take a spine to do this. And so this first picture here shows how we take a little section of their spine. It looks a little gruesome, but you know, those 90 fish that we've recaptured, we've seen how they healed, and they actually heal pretty well. They grow right back over. They're not as strong as they were the first time, but they're still really good. What's cool about these spines is that they put rings down just like a tree. And so we can count those rings, and it's a pretty good estimator of how old these fish are. And so, you know, here's an example of what it would look like here. This was a staged photo of me estimating the age of that fish. And so with those ages, we're able to do a lot of, we're able to answer a lot of questions. And so the first question was, when was that fish born? And so that's what we call this cohort year. And so this figure here shows the year that a fish was born, and it shows out of all the fish that we caught, how many were born in that year. And so when you look at this graph, you can see there's not a lot of fish that were born in the 80s and 90s. So those would be pretty old fish. So there's not a lot of old fish. Suddenly in 93, they start really going up, and then they come down in 2005. And then here, there's not really a lot of young fish. And so this was really interesting to us. We didn't know, we don't think that this is normal. We sort of expected there to be a little bit more old fish, and we're a little worried that there aren't young fish. So when we looked at each year that we ran this survey, so this figure here shows the year of capture, and then it shows a plot of all the fish ages. And so in 2010, the fish ages ranged from 5 to 30, 27, I guess, right? So that's what this plot's showing. And when we look at each year, we noticed that our population is increasing by 1 year each year. And so that's, again, that stood out to us as sort of weird. We didn't know what it meant, and we thought about it. And, you know, if you think about a population in equilibrium, you'd imagine there'd be the same number being born as the number being dying, either from old age or from natural cases. And so you would expect that line to be flat. If the line was negative, that would be the fish were they were not being born, and they were dying. And so what we are seeing is a positive slope on that line. And so what we're thinking is that the fish are getting older, they're not dying, but there's no young fish coming into the population. And so one of the questions people posed right away was, well, does your gear only catch old fish? And so we wanted to look at that. And so we looked at each year, we looked at the age distribution of the fish, right? So each one of these graphs represents a different collection year. And so what we realized is no matter what year we collected fish in, there's very few fish after 2002. And so this is kind of a hard thing to explain, but in 2010, if we caught a fish that was born in 2000, it would be a 10-year-old fish. In 2015, if we caught a 10-year-old fish then, it would have been born in 2005, right? So we know that we're able to catch 10-year-old fish because we did in the past, right? We caught 10-year-old fish. So why aren't we catching 10-year-old fish here? It sort of answered the question that we're not biased in our collections. We're doing the right stuff. We're doing the right tool, but the fish just aren't there to be caught. So that was really concerning to us. And it sort of led us to this next question. We wanted to find out, okay, all these fish that are out here, are any of them sexually mature? Are any of them reproducing? So we had this whole question about, you know, reproduction. And so one of the challenges with lake sturgeon is that they're not easy to identify their sex. They all look the same. The only way is to see their reproductive organs on the inside. And so this bottom picture, we would perform a surgery and try to look and see if we could see whether it was a male or a female. And this top picture here, we were doing hormone analysis, trying to see if we can do that to see if they were male or female. And so all these things was a lot of effort. And just last year, somebody figured out that you could sex them genetically. And so all the effort that we did back then is just right out the window, and we have a much easier process now. But the best part of this activity was in 2012, we observed the first sexually mature female that was ever documented in the river. And so that was a really exciting day. And so these are some of the eggs. They're this black gold color. They're an amazing color. So it was really a big deal. And so once we started seeing sexually mature fish, the number kept going up and up and up. And so, from that marker capture study, we knew we weren't catching all the fish, right? We were really bad at it, actually. And so when we see 12 mature female in 2018, we were really excited. We think, yeah, okay, they're really starting to reach the age of sexual maturity, and they're really, really going to take off here. And so we started monitoring their spawning. We put out egg mats, and like I said, these eggs are sticky. And so they would hit these mats and they would stick to them. And so we caught hundreds of eggs in over the last couple of years. And here's our little fish. I think we named him Stercules because he was the one that lived the longest. But we hatched them out just to make sure that we were in fact looking at sturgeon eggs. And then, so, these were sort of this stepwise understanding of the sturgeon. We know they're sexually mature. We found their eggs. We hatched them out. They're healthy. Then we did larval sampling and we found that they were surviving. So we've got evidence now that that reproduction is not the problem. And then the best part was we actually got to see some spawning behavior. And so here's a video that I took from the shoreline of the river. We were right on a hiking trail. And in this video, there's probably 15 or 16 lake sturgeon, and they're just spawning in about 2 feet of water. Now, in general, this is just amazing to see this many of these fish after they've had such a rough time population-wise, and to see them spawning like that. But then you also look at them and look how vulnerable they are. They're in 2 feet of water, and people used to just spear them from shore and collect them for food and stuff like that. And so these are probably all 30-year-old fish that, are susceptible in that way. And so it was really exciting, but also sort of, oh, wow, kind of moment. So from there, we had some questions about their habitat. Where are they spending the most time? And so what we did is we used acoustic telemetry. And what that means is we put tags inside a fish that emit pings, little sounds, and we have receivers out in the water that end up listening to them. And so they act sort of like checkpoints, like easy passes. The fish swims past it. We know the fish number. We know the date and time that they swam past it, and we could then start looking at where they're spending the most time. And so here's an example of a receiver. It's on a big, heavy concrete block. We throw it out in the water. It's underwater, and then we have to recover it. And so once we recover it, we can download it. Here's in the background, you can see the river. It's quite high flow right there in that section. And so here's a picture of us implanting the tag in the fish. We did this surgically. We would use an anesthesia to knock the fish out. Here's a coworker putting the water through the fish's gills. And here's the surgeon. It looks like he's buttoning up this little hole here where we put the tag in. And so we have this great site. This gentleman let us use his shore house, his boathouse here, and we would do our field work right there. This is an image. All these black dots represent the receivers that we put out there to track where the fish is going. And so again, this is where the fastest water is. This is where we think that they're spawning. So we had some receivers up here. That's as far up as you could go by boat. And then we had just monitoring for the rest of the area. And so this is an example. This is one of the cool things you can do with the data. You can just plug it into this package that puts it into a video. And what each of these colors are is a different fish. You know, we tagged 85 fish, so there should be data here from all 85 of those fish. And all the black dots are the receivers hearing where they would hear them. And so what I want you to notice is just how these fish move around and where they might cluster. And it's sort of one of the take homes on this is it's kind of crazy how much they do just different things. They're so individual, right? And so here you can see they're all just moving around. A lot of them are out here in the lake, but a lot of them are coming up here to spawn. This period doesn't really line up great with spawning. I couldn't get a good section of it, but you know, at any given time, there's fish in the river, there's fish in the lake. They're sort of all over the place, right? But they don't go very far. They're all sort of staying around here. We have receivers out here, and you'll see some of these kind of bounce back and forth, but a majority of them are staying around here. And so from this data, we take this data and we put it into some statistical models to try to think about where they are and what they're doing. And we actually can make sense out of it, believe it or not. And so one of the ways we look at it is how long is a fish being in our array, right? So we have all these receivers out there. How much of the time while they're out there are they actually in the array? So each one of these lines is a fish, right? These are all fish IDs. And if there's a black dot, it means it got detected that day in our array. And so when you break this up by season, you can see in the spring, in the summertime, they're usually in our array. There's not too many of them disappearing. And so we have 90% of the fish are in our array. In the autumn, it drops down a little bit. You can see over here, they start leaving. It's down to 70%. In the winter, they really disappear, right? And so I was talking about that overwintering habitat. Our array doesn't capture that overwintering habitat. They clearly are leaving and going somewhere else that we don't know because we don't have the receivers there to track it. And then again, in spring, you can almost see right in the middle of spring, they start coming back for spawning, right? They all sort of pile up here. And so we're starting to see seasonally where these fish are moving around. Another thing we can look at is when they come in to spawn. And so what we have here is, this is date on the bottom. It covers about 3 years of time. The blue line is the temperature, right? It goes up in the summer and it goes down in the winter. And so that's each year. And this red line is showing the number of fish that are showing up in the spawning area. And so they sort of, there's 3 peaks here, and all those peaks line up with 12 degrees centigrade. So they're clearly coming in to spawn at a certain temperature. Like that's the big driver in them coming in. And so we were able to look at that with this telemetry. I already sort of talked about that. This is the residency index over time. And so the fish are in our array and they leave in the winter. And so we're seeing that repeatedly happening. And then this was another figure that just shows hot spots, right? And so you can remember from that video, I mean, most of the fish were just out on the lake right near the mouth of the river, right? And so we've got a lot of these hot spots here. And so we've been able to identify some important habitats. Home range size is another thing that we looked at. So this is, you know, how far do they move from within like a period of a month. And so in the winter, they're not moving a lot, but in the springtime, when spawning starts, they're making big movements up and down the river system. And so ultimately, we take this data and I'm trying to get back to that habitat use triangle. And so we've identified where they're breeding. We've used the telemetry to find those points. We know when they're doing it, and we've collected eggs. So we know pretty good spots where they're doing it. We've identified that they do a lot of feeding ground. They spend a lot of time right outside the mouth of the river, where we think is, big time feeding grounds. It's really sandy, a lot of fish and mussels and things there for them to eat. But we still haven't identified where their overwintering habitat is. And so that's something that we're working on in the future. And we've also seen that this is, the river itself is a migration corridor. They'll come up and down using that river. Excuse me. So I think I'm running a little, I think I'm good on time. I was going to skip this section if I was getting short, but another thing that we looked at was what they're eating. And so to do this, it's funny, it's called gastric lavage, but what you do is you pump water into the stomach of the fish, and then you push that water back out, and it sort of washes out what's in their stomach. And so by doing this, we've collected all these different prey items of things that this fish was eating. And so up here, these are a bunch of amphipods. It's a little hard to see, but there's a big fish in these bottles. In here are some crayfish. And so it was really neat. There's all sorts of things that were coming out that they were eating. And so we did two different ways of looking at it. We looked at the stuff that was in their stomachs using stomach content analysis, but we also looked at stable isotopes, which is a chemical signature of what they're eating. And so when we use them together, we get a really good picture of what's going on. And so there's three ways to look at what's in their stomach. You can look by how often it's in the stomach. You can look at the weight, and you can look at the count. But in general, they all sort of point at these two critters being the most important, the amphipods and the round gobies. When we do the stable isotope, those chemical analysis, we're looking at carbon and nitrogen isotopes. The carbon helps tell us sort of where the source is. It says where the primary productivity is, but the nitrogen tells you sort of what trophic level it is. Are you eating the grass, or are you eating the thing that eats the grass, right? And so it sort of helps identify where you are in the food chain. And so here's just an example of collections. We did some baseline collections of what's out there. And then we took a little piece of tissue from the fish, and we took some blood from the fish and analyzed them. So I'm just gonna show this quick. This is the carbon chemical signature from left to right. These numbers don't really matter. It just shows you where the different organisms fall that are out there for them to eat. And then the nitrogen shows you what trophic level they are. And so essentially, if you're up above one of these organisms, you're most likely eating that organism. So the crayfish are probably eating these guys. The round goby is eating crayfish. And then up here, these are the sturgeon. And so the sturgeon are most likely eating the round goby, according to this analysis. And so those all sort of lined up with what we were seeing in the stomach. And so that was really interesting. I think I get into it here in the summary, but what's interesting about those amphipods and the round goby is they're both invasive species. So almost all of the diet of this sturgeon is eating invasive species that have replaced whatever native food source used to be here. So to summarize what I've sort of a big pile of stuff that I threw at you today, but we looked at these fish and we counted estimated how many were out there, and there was a lot more than people thought. We saw that there was that one period where there were really strong year classes, but then it's sort of, there wasn't old fish and there weren't young fish. And so that was something that we're sort of concerned about. But we've seen reproduction happening. We believe that they're surviving, and we've started seeing more and more spawning fish. So the big story on this population in the lower Niagara River is that they're on their way towards recovery. It's taken us 8 years to collect all this data, but we really have enough evidence now to say that there's nothing that should be holding them back except for time. It's going to take a lot of time for them to keep restoring and reproducing. I mentioned the feeding, the invasive species, and some of the seasonal changes that we've observed with that telemetry stuff. And so in the future, we're still working with these fish. We still have the telemetry system going. And one of the questions we want to pursue is whether what are the actual rates in this population of how often they're coming back? Are they reproducing every 2 years for the males and 3 years for females? And so we're going to answer that. We're also want to look at the lake-wide movements. Where are they going in the wintertime? Do they go to other rivers to spawn? In that video that I shared of the dots moving around, we saw probably like 4 or 5 of them take off in some direction, and we don't know where they went. So we really want to learn more about that. So that was a lot. And again, I appreciate your time. Thank you. And I guess let's see what kind of questions we might have.
Yeah, thank you. That was great.
Yeah,
thank you so
thanks.
much. Love all the pictures. We do have some questions. Before you get to the questions, there was,
Okay.
Caitlin asked when you have time, if you could just go back to the slide with the causes of decline.
Yeah.
See that again.
You bet. You bet. It's somewhere. We'll get there. Did I pass it? There it is. All right.
So this, before I'm going to squeeze my question in since I have the floor. So you said that the round goby are invasive, and it's one of the reasons for their declines, but it's also one of the main things that they consume. So how does that make sense?
So we're pretty worried about that because they eat eggs, and so they could interfere with reproduction, but they have such a benefit in the diets. Like our fish, once they started eating round gobies, started growing faster than like other populations that didn't have round gobies. So it's kind of tricky, and we're not sure which is the bigger impact. I think the benefit that they give to growth outweighs whatever they're doing to the eggs. Yeah, and so it's a little tricky. And we also don't have like concrete evidence that they are eating the eggs. I mean, it's just sort of assumed, like that's what they do. So we don't know like how much they eat or anything like that.
Great, thank you.
Yeah, I have a quick question. Did I convince you that sturgeon are like the most awesome fish ever?
I think I'd be afraid if I saw one in the water with me.
You got a couple of thumbs up, so that's good. All right.
I go in the Lamoille River. I don't really want to run into one. Do you want me to go through the questions, or do you have them pulled up there?
Yeah.
All right, so you might have answered some of these as you went along, but we'll just kind of start from the top. So are there more? This is kind of, I think, in the next set of data that you were showing about, are there more fish, or are you guys just getting better at catching them? So I think you answered that you're actually not good at catching them. So that wasn't the reason.
Right. So we're not really good at catching them, but we did get better, but every time we catch fish that are new, it just keeps bumping the population number up. If we got really good at catching fish, we would just keep catching recaptures, and then our population would get we would really like hone in on a really good number or an accurate number. But in our case, we keep getting new ones. We just get so many new ones. And so the population just keeps, the estimate keeps going up. And so the next question is, would we rather recapture them or get new ones? I think getting the new ones is better. It's awesome because that means the population is bigger. It just keeps increasing the population number. But the way the model works is we need a lot of recaptures so that our estimate, the error bars around our estimate is better. So it's kind of two different reasons for wanting both. So the next question is, how did we determine that this is how to get their age? We didn't do it. Somebody else did it a while ago. There's a lot of different fish. A lot of people do this on different fish. Some fish, you can do it with their scales. Their scales will have rings like a tree. And other fish, you have to use one of their bones or spines. I think back in the 80s, somebody did this really interesting study where you can take each of those rings and analyze the amount of radioactive carbon from like nuclear testing, right? And so all the nuclear testing that used to happen, there's like levels of radioactive carbon, and it decreases over time. But that signature is in each of those things. And so you can validate that each, this fish, is born at this time because in the air, that's what the radiation level is. So it's kind of crazy, but that was probably the study that really showed how to do that. Fewer fish from 2002. Yeah, that's when I had my big break. So there is a conspiracy to that. Yeah, I guess I stole their thunder, and then I came in 2010 and brought all the fish back or something. I don't know. What is the birth rate? That's really hard for this population. And so to get the birth rate, you'd see how many eggs are laid and then what the survival is. The water where these fish spawn is so hard, we can't get a good number on how many are laid. And then we struggle to get a number on how many then hatch. There's some smaller rivers in Michigan that have done a great job of doing this. And it's surprisingly low. It's like 10% maybe. So only 10% of those eggs will hatch and become little guys. And then those little guys even have a harder time. So fish survival is, egg survival is really low in general. And that's why they lay so many eggs. That's why they reproduce so often. When you look at these sturgeon, they chose to get big and fat and full of eggs. They'll lay like 10,000, 20,000 eggs. And so that's all in hopes of one of them surviving, right? And that would be successful. So birth rate is pretty low for fish. Was the gender searching hurting them? No, we anesthetized them. So they were sort of knocked out, and we didn't do a lot of poking around. Usually you could just look in that hole pretty quickly and see what it was. And, I imagine it's just like us getting a surgery. We're probably a little sore afterwards. But we did our best to be, really sterile, really safe with the fish and do the least invasive stuff that we could do. Oh yeah, do they go right back in the water or do they need to be sterile? So we suture them back up and we make that, as good of a tissue to tissue getting back together. And then we put iodine on there to make sure that it, any surface bacteria is gone. We'll leave them in that sling for a little bit, get in fresh water, and they'll recover a bit there. But they do then go back into the river. And it's sometimes not the cleanest river. I mean, the Niagara River has a couple sewage treatment plants on it, and it's a lot of chemical contamination. But we have had really good survival. All those tagged fish, 85 fish, we only have one of them that we think might have died after handling. It was like a month or so later. And so that's pretty good survival for the work that we were doing on them. Yeah, so the females have longer lives. It's so that they can reproduce more. The goal is for them to reproduce probably 10 or 15, 20 times in their lifetime. And the males are just, swimming around trying to find the best females out there. It's kind of like the bar. So one of the causes of decline is water pollution. Are sturgeon more sensitive to water pollution than other fish? That's interesting. I'm not sure how to answer that. I think the sensitivity of the fish to pollution, I think they're all sensitive to pollution the same way, but I think it's how they eat, what they eat, and how they absorb that pollution. And so you could have, a minnow that goes around and eats little tiny pieces of plants, and that might not pick up the same kind of pollution that the sturgeon pick up when they're going around eating, dead fish off the bottom or mussels or things like that. And so the pollution will have a bigger impact based on how you feed and how long you live and things like that. And so it's not that they're more sensitive. I think it's just that they're more susceptible to the impacts of it. Okay, can I go to my research side? Okay, is that the questions, I think? Yeah, okay. Okay, so I got a question from Robert about do we catch other species because we're not the most efficient? We used to catch catfish, and we had some trout that we've caught sometimes, but we started figuring out where to fish that we didn't catch those other fish as much, and we were just catching sturgeon. They're also really big hooks, and so a lot of the smaller fish weren't getting hooked on them. And so, we call that bycatch, and we really didn't have very much bycatch. It was kind of lucky, I guess. But it might also mean that our methods aren't good. I think that's what you were asking. But the alternative, I don't know if you're familiar with gill nets, but they're essentially, really fine material like screens that you stick in the water and the fish get stuck in. That's the other option for catching fish. And we've tried it, but there's so much stuff in the water, so much vegetation and stuff that it just clogs, it gets on these nets and completely ruins them. A lot of researchers use that instead of the set lines like we do. And that seems to be a bit more effective, but we tried it and we just couldn't do it. How do you know the rings determine the age of the fish? I guess I answered that before. Is that how you found the age of the oldest fish? Yeah, so I'm not sure what this means. The rings do not stop growing after a period of time. Every year, they add another layer to the outside ring. And so they just keep growing out from the center. And so, as long as you can find that center, you can keep counting the rings all the way out. And honestly, once the fish gets past like 20 years old, it gets really hard to count the rings because they get really close together. And you might be off by 5 or 10 years at that point. If you're looking at like a 70, 80-year-old fish, it's very difficult to do that. And so it's not 100% accurate. I don't know if I would say no, taking the spines does not affect their lifespan. I think they grow back, but they don't, something like that wouldn't really affect them unless they got an infection or something on it. I think if they got infected, you could have some bigger problems down the road. For the most part, the fish that we recapture look really good and really healthy. They don't lose weight. They continue to grow. So I think that they're in good shape after we take those spine pieces. So what would I say the main point of what I've shared today is, well, so I said it before, I think I wanted to convince you all that sturgeon are the most awesomest animal in the water. So if I did that, that's a good takeaway. But the bigger takeaway is I feel like it's really hard to find examples of positive change in nature, especially with fish. I think, you see things with birds a lot. There's a lot of good stories of recovery with birds, but with fish, it seems to be pretty difficult. And, I want what I use to sort of motivate me in my work is that I want our impact on our waters and fish to be negligible, right? I want the fish to be able to do their thing and to reproduce and grow and not have problems because of something that we want to do in our life. And so I feel like this is a good sign. It's a good indicator that we're doing the right thing. The regulations that cut down on, the Clean Water Act was a good move that, started helping recover sturgeon. Any habitat improvements that we can do. I talked about how we destroyed habitat and how we put dams up. Well, now people are taking dams out and they're actually putting rocks back into the river in the sections where they cleared them out. And so those actions are making a difference. And so I guess that's my takeaway, is that our actions can make a difference for good and bad. And so that's a good point. How many eggs are produced by the females? I think it's between 10 and 20,000 eggs for each female. And I said, it's under 10% of them actually survive. There's another question about the spines showing age. Yeah, okay, internships. We do internships, and somebody has to go to the life cycle slide. Let me do that, and then I'll talk about internships. So we do offer internships. And of course, COVID really threw that into a lurch. We haven't had interns for 2 years. I don't know if we're going to try to do it this summer, but what I can do is I can send Dr. Tarleton the information for any internship opportunities. I would be more than happy to have any of you out there. I think, a very real challenge would be that they're not paid internships. So, you'd have to come out here and that wouldn't be easy. But with that said, I could also work to make a connection with the state people that are doing work on the Lamoille, if that would be interesting to anybody. But yeah. So,
We can have them come talk at this class next year. So happy
oh
to
yeah,
get in contact.
yeah, that would be great.
So we're just about out of time. Thank you all for your great questions and for following up on clarification on some things. That's always helpful. And thank you, Dr. Gorsky, for being here with us today and taking time out of your schedule to share your important work with us. And we really appreciate your time. Thank you again.